I'm just going to be going over the Calvin cycle and the light reactions for lab. I apologize in advance for the animals. Cat's over there somewhere. There it goes. They make a lot of noise. I can't control that. I'm sorry. So first I'm going to be going over the Calvin cycle. And from what I remember from botany, the Calvin cycle is basically kind of what plants do when there's not enough available light. They kind of just kick over into the Calvin cycle in order to, continue to make food for the plant. And the end goal of the Calvin cycle is to make a molecule of G3P. And in order to make just one molecule of G3P, the cycle has to run through at least 3 times. And it takes at least 2 G3Ps to make a whole entire molecule of glucose. So that's a total of 6 times just to make one molecule of glucose. I'm sorry. Y'all stop. Okay, but so the Calvin cycle is broken up into 3 stages, 1, 2, and 3. Stage 1 is the carbon fixation stage, which is this portion. Stage 2 is the reduction stage, this portion. And stage 3 is the regeneration portion. So starting off with stage 1. So 3 molecules of carbon combine with Rubisco. It's like an acceptor kind of molecule in order to make 6 molecules. I can't pronounce these words, so I'm just going to use the acronyms. So in order to make, the carbon has to combine with the acceptor in order to make 6 molecules of the PGA, the 3-PGA. It's like some sort of phosphate thing. And then while this is going on is like a side product of the reaction, 6 ATP molecules used in this this portion right here is converted into ADP, which will be necessary later in for getting into the NADHP or the NADP down here. But so then after that, moving on to stage 2, the reduction stage, the ATP and the NADP are used to convert the PGA molecules up here into the 3 carbon in order to get into the G3P. So after coming down here, we need to get into the G3P. So that's where the ADP comes in handy over here in order to get to our end product over here. And as a so it donates 6 electrons, so as a product it gains 6 protons, becoming more positively charged because it loses those 6 electrons. Then moving on over here, the regeneration stage, some of the G3P molecules will go down to make the glucose, but then some of them have to be recycled in order to regenerate this receptor up here, the Rubisco, so we can start the cycle back over again. So losing, one of the molecules, starting out with 5 over here, the G3P will go all the way back up here, kind of like reversing in a way what happened over here in order to get back to this base starting point up here. And in order for one of the molecules to be able to leave the cycle, more carbon has to come in, or else it'll just, just sit there and just go around all day. So another outside factor has to come back into the cycle in order for a stage to exit. And this is called, the regeneration portion, because we're regenerating the Rubisco acceptor in order to start the process all over again to make more G3P. Okay, moving on to the light reactions. This is like what normally people think of when what happens with plants, like with normal photosynthesis and things like that. Light energy is introduced into the stroma, I think, of the chloroplasts in order to kickstart this whole process we're about to go through in order to make molecules in order to be transferred into sugar. So the light reactions always start out in photosystem 2 with the P680. I guess it's like just a pair in the photosystem is what I've read about it. So when light energy reacts into it, it excites these photosystems, and it causes it to become an electron donor, and it'll donate these electrons, moving from a positive to a negative energy rate, because it's donating the electrons. So it'll go up in the energy combined with things like water. Breaks down into these different molecules into the electron transport chain. So when it gets excited and goes through there, it's transferred into all these molecules. I can't really say these words, but phenylalanine and then plastiquinoline, which is then transferred down into the cytochrome B6 complex, and then as it moves through the transport chain, becomes the plastic. Different from that word, but plastiquinoline again, and ATP is generated here as well as sort of like just an energy releaser, like an extra. And then, still moving on with those 2 electrons, we come down to photosystem 1. And photosystem 1, it goes back down to that higher energy grade, the P700, because we've lost the energy as we've gone down through the electron transport chain. So now it needs to be regenerized, and that's through the absorption of more photons. So going down to photosystem 1, light energy excites the molecules again, or the electrons again, and it transports back up. And then this time, the end releaser, it releases, it's turned into some proteins, and then like specific proteins, ferredoxin, and then it's transported into an enzyme reaction that builds NADP reductase, which then transfers electrons to the NADPH carrier that are used to build sugars from carbon dioxide, like in the Calvin cycle. And so without this process going on, the Calvin cycle wouldn't be possible, because even though the Calvin cycle doesn't require light energy specifically, it still needs the molecules that are created using the light energy. And this is just showing that the direction of the energy. So yeah, that's both the cycles.